Okay, here's what we're doing today. We finished before Christmas, and most of you guys did a really good job on the quiz that we had, but we finished before Christmas with DNA and the three functions of DNA, or the three things that DNA does. So we are now getting into RNA in translation and transcription. So by the time we're done, maybe not today, but later on this week, you'll know how a DNA message can be transcribed into a message, actual instructional code in the form of RNA that can then be decoded into proteins that work to do certain things, like fight viruses. So when they look at the vaccine that's given for the COVID-19, the coronavirus, it's actually an mRNA vaccine. And we'll talk a little bit about what that is too. What was that? Okay. So you guys will learn the role of RNA, and then we'll talk about the differences between RNA and DNA. And then you should be able to describe the process of transcription and translation. So you are going to have to open up a new tab and join this Pear Deck. Join this Pear Deck. Joinpd.com. Oh, let me put the code up there. And then there's the code HDGHXB. All right, about 30 seconds more. Got 3 more students we're waiting on. Let me know if you can't get in for some reason. All right, one more. Who's not in still? 10 more seconds. Okay, so listen, the first part of this is actually comparing RNA to DNA. Between those two strands, each of those is still made up of that 5-carbon sugar. The only difference is between the sugars is that DNA is deoxyribose and RNA is ribose. But it's easy to remember that because RNA starts with an R and ribose starts with an R, and DNA starts with a D, and deoxyribose starts with a D. So there's actually 3 differences between RNA and DNA. The first one is the sugar type. They are both made of a 5-carbon sugar, but the difference is the names of them. RNA has ribose instead of deoxyribose. Remember that DNA was double-stranded. We call that the double-stranded helix or the double helix. RNA is single-stranded, so that's another difference. And then when we match the base pairs, the nitrogen bases, A was going with T with RNA, we now replace T with a U. In other words, when we transcribe DNA to the messenger RNA, A's will now be matched with U's instead of A's being matched with T's. So it used to be ATCG. Now it will be AUCG. AU, A. And these chemical differences, they make it easy for the enzymes to tell the DNA and the RNA apart. Understand, too, that the messages that we're creating, the mRNA, there's actually 3 types of RNA, but the message that we create, the first step, is formed from the DNA. And I like to use the analogy of loading a roller coaster before the coaster takes off from the place that a roller coaster takes off from, where they load it, the loading dock. So I'll show you what I mean once we get into it. Okay, so here are the comparisons again in a little 2-section notes here. DNA is double-stranded compared to RNA being single-stranded. The 3 parts of DNA are the phosphate group, the sugar deoxyribose, and the base is A, T, C, and G. And then the 3 parts to RNA is a phosphate group also. This time the sugar is ribose, and the bases are A, U, C, G. So you can see the differences once I get this mouse out of here. Down at the very bottom there, underneath the presenting, where the differences in the base pairs is that DNA contains thymine, the T, RNA contains uracil, the U. All right, another little 2-section notes here, breaking it down. Again, these are good notes to look at because it shows you all those differences and comparisons between DNA and RNA. Okay, so here's the role of RNA. Your genes, my genes, our genes, contain DNA instructions that actually tell our cells how to build proteins. Remember, proteins were one of those major macromolecules, and proteins are the polymer of smaller amino acids put together. So the first step in doing this is to take a sequence of DNA in the nucleus and decode it into an RNA strand. RNA is a nucleic acid also, and it consists of long chains of A, U, C's, and G's, the nucleotides. This RNA uses the sequence from the DNA to send out a message that has the code for producing proteins. And again, remember, proteins have like a lock and key feature where the shape of the protein's got to lock into whatever function in the cell that it's performing. So building these proteins with the exact order of the amino acids develops different proteins that do different jobs. And again, it's actually a pretty cool process when you think of all the things that are happening in just a little cell. Okay. So the cell uses the original DNA strand, just like 2 weeks ago when we were replicating DNA. It'll take the original DNA master strand and it'll make a message out of that into RNA. The DNA replicates inside the cell's nucleus, while the RNA goes to different parts in the cytoplasm. The ribosomes, remember the ribosomes are the assembly crew. That was me again working at Harbor Steel, punching steel plates. But the message from the RNA actually goes through the cytoplasm to the ribosomes to put together a chain of amino acids, a polypeptide chain, that the order of those amino acids will form a certain protein. Okay, so just take a guess as to what you think the enzyme is that's responsible for adding these nucleotides. Take a guess here. Your choices, remember we talked about the helicase, the polymerase. That might have been the two we talked about, the helicase and the polymerase, when we were talking about DNA. So during this DNA replication, A pairs with T and C pairs with G. Oh, this is review. So helicase and polymerase. One of the enzymes was used to unzip the DNA. The other one was to add the new strand. So this is during DNA replication. Remember back then, A paired with T and C pairs with G. So they're just asking what enzyme. So take a guess. Your two choices are helicase or polymerase. You could put H or P if you want, if you're not sure how to spell them. Okay. So that was actually the polymerase that added it. The helicase is what unzipped it. So now, going from DNA replication, the DNA is unzipped. Before it replicates, it can make a message of itself through RNA. That's called the mRNA. So most of the work of making RNA takes place during transcription. There's two parts to forming a protein. There's transcription and translation. Most of the work takes place during transcription. During the transcriptions, we take a segment of DNA, and that serves as a template for the RNA. The bases of the transcribed RNA complement the sequence of the TNA, of the template DNA. So look at this right here. Look at this right here. Here is your original DNA strand, T, C, and G. Your message RNA will pair with that original strand. So the message with T, C, and G would be A, G, and C. Look at, though, the last three letters there, ATT, because now instead of T matching up with A, we add U to it. Here's where it becomes a little bit confusing, is that A still pairs with T, but U pairs with A. So let me show this again. This is just enough so it's not covering that bottom part. So we're looking at this DNA strand right here. And what we're doing now is we break the DNA into three letters. We call those codons, three-letter codons. So the first three letters, the TCG, the message is the AGC. A pairs with T, G pairs with C, C pairs with G. The second three-letter codons is GTC, where C pairs with G, A pairs with T, and G pairs with C. And then the third three-letter codon is ATT. But now, notice that RNA cannot have any T's in it because all of the A's are paired with U's now. So again, RNA cannot have any T's in it, and DNA cannot have any U's in it. The RNA polymerase is the enzyme involved here, and it occurs in the nucleus. So this is the actual loading of the roller coaster. And here's what I mean by that. If you go to Michigan's Adventure or Cedar Point, there's usually a teenage kid working the dock at the corkscrew, the roller coaster, the Shivering Timbers. The one that goes straight up to the top and then comes straight down. Thrill dragster, the dragster. Anyways, it's usually a teenage kid that you probably don't trust that much to load you onto a roller coaster that's going to shoot you upside down. But that's what's happening with the RNA also. The RNA is loaded into a roller coaster. Okay? The DNA has the initial code for it. The mRNA, the message RNA, are the actual people getting to the roller coaster. Once that message is loaded or transcribed, the teenage kid working the dock puts his thumb up, and some other teenage kid pushes start, or however you start a roller coaster, and it leaves the dock. The same thing happens with the messenger RNA. Once this message is transcribed, that's transcription. Once it's transcribed from the DNA, whoop, it leaves the nucleus and it goes to different parts of the cell. This through the cytoplasm, and it finds ribosomes. Okay, so listen, try this one. I think it's multiple choice. Transcription, then, the only thing we've talked about so far is transcription. Transcription is the process of converting something to something else. Just remember that the message always starts with DNA. That'll give you the answer. The message always starts with DNA. So transcription is transcribing the message from the DNA. Good, good. Okay, so transcription is the process of converting DNA to RNA. That's what you should have put there, converting DNA to RNA. And then the enzyme involved in transcription. Remember, they only had you focus on one enzyme here. I think this one's multiple choice too. The enzyme involved in transcription. Good job. Good job. Just take a guess on this. Okay, so listen, look at this screen right here. This is pretty good. So here's your original DNA strand. Here's your original DNA strand. The DNA enzymes, helicase, unzips it, and then the RNA polymerase forms the message. Now notice once the message is formed inside the nucleus, whoop, it leaves and it goes to different parts of the cell. Okay, so load the roller coaster, give the thumbs up, the message leaves the nucleus and it goes to different parts of the cell. Okay, so here's where your genetic code comes into play. The genetic code are the billions of ATCGs that we're made up of. The order of these ATCGs determines the amino acids. So RNA contains 4 different bases: adenine, cytosine, guanine, and uracil. These bases form a language, and it forms those language with 4 letters: A, C, G, and U. So here's your 3 types of RNA. There's 3 types of RNA. I briefly just went over the messenger RNA. There's also a transfer or a tRNA. And then there's a ribosomal RNA. That's where the packaging takes place. So again, the 3 types of RNA is the messenger. That actually takes the message from the nucleus to the ribosomes. Once it's in the rRNA, the ribosome, that the transfer takes place, where the tRNA brings what we call anticodons with the actual amino acid. Okay, so here's your genetic code. Each 3-letter codon, we call them. A codon consists of 3 letters, helps to form the message from the mRNA. So if you look at this right here, there are 3 3-letter codons. The first one is AUG. The second one is AAC. And the third one is UCU. Those three-letter codons will actually bring 3 amino acids to that message. And that's the translation part, is taking the message, the mRNA, and converting it into a protein by adding amino acids. The order of the amino acids determines the protein. So here's how this three-letter code is deciphered from this chart right here, this wheel. All right, do this. Do this if you can. This is how this wheel is read. And I'm going to stay up here because when I move my mouse, that presenting thing comes up. So I'm going to stay up in this section. Let's just say that our three-letter codon was GCC. Our three-letter codon is GCC. So what I would do to figure out what the amino acid is I start with the first letter, the biggest letter in the middle, G, and then I follow it out to the outside of the wheel. My codon, my three-letter codon again was GCC. So it's going to be G, C, C, and my amino acid is the alanine. Now, alanine can have 4 different codons for it. Alanine can be GCC, like I just said. It can also be GCU, GCA, and GCG. All right, do this in the chat. Do this in the chat. Tell me what the amino acid would be for GGA. In the chat, the amino acid for GGA. Remember, start on the inside with the G and follow it out to the G and the A. And then on the very outside, you'll see an amino acid that starts with the letter G. Type that in so I know that you guys are following still. Good. Andre, don't transcribe it. Get the actual amino acid. That's the word on the very outside of the wheel. That's a good job, you guys. That's a good job. Okay, so look at GG, what did I say, GGA or GGC? It doesn't matter. It's going to be the same. So if I start with the G right here and I follow it out, the only amino acid, if I go GG, is going to be glycine. So that's a good job. All right, try this one. Try this one. I'm going to go to the bottom part now. Do this one in the chat. Do CCC. CCC. CC, pick up that guitar and talk to me. Do CCC. All right, good job. Good job. Yep, I think you guys got it. You should find proline. You should have found proline. So again, if you look at that right here, CCC is proline. When you guys come back next week, we're gonna play a little codon bingo, where I give you the 3-letter codon and you have to determine the amino acid. All of these amino acids put together in a specific order are what form the protein. That's what forms the polypeptide chain that determines the protein. Oh, do this one. Here's an actual pair deck. AGC. AGC. Follow it from the middle out. Good. Good. AGC. All right, so the 6 kids that answered, we've got 100% on it. 700%, that's good. That's good. So we're getting it, that's good. Oh, try this one. Try this one. What amino acid does the codon UUA code for? Looking good. Couple more responses. Looks like 100% so far. All right, good. It is leucine. So UUA, UUA, and that's the leucine. Oh, one more. CUC. All right, good job again. Good job again. Now remember, that's why we were saying sometimes you can have more than just one 3-letter codon code for the same amino acid. Leucine has a bunch of at least 6 3-letter codons that code for it. Okay, so that's what this is saying right here. Most amino acids can be specified by more than just one codon. There's 6 different codons that code for leucine. But there's only one codon that codes for the amino acid tryptophan. There's only one that codes for tryptophan. Oh, try this one. Try this one. AUG. AUG. Good, good. You guys got this. All right, it is methionine. Now, methionine is I'm going to skip this. I'm going to skip these. Methionine is an important one because that is actually the start codon. Whenever you have an AUG, that methionine serves as the start for the protein. There's also stop codons, and those actually say stop on the wheel. There's 3 different ones that serve as a stop codon. But anyways, the methionine, the AUG, that starts the protein synthesis. That starts putting the amino acids together at that point. Okay, so here's just the review of what we've covered with transcription. Transcription is the process from going from DNA to RNA. The original DNA strand splits, and once it splits, the message is transcribed into mRNA. So let me show you this right here. Okay, I'm going to share this. Okay, watch this right here because this is the transcription taking place in the cell. Here's your original DNA strand. Here's your original DNA strand.
DNA in the gene is used as to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand.
Okay, so listen, this is that part. This is the original DNA strand. This is the original DNA strand, and it's split. This would be loading the roller coaster. So they take one part of the original DNA strand and they form the mRNA, the message from that part. So loading the roller coaster, once this is all loaded, this message is going to leave the nucleus and go to different parts of the cell.
As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped.
Okay, so watch this real quick. Watch this real quick. I've got the message up here. This is the mRNA that was formed. But watch how the amino acids are brought to it. That's the tRNA. And I'm kind of getting ahead of myself because I'm going to stop here in a minute. But I just want to show you how those 3-letter codons help with the tRNA bringing the amino acid.
Grouped into 3-letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific.
Hey, this is it right here. This is it right here. So AUG is the start codon. There's actually an anticodon that pairs with AUG that brings the amino acid to the correct spot. So AUG would be UAC, and that UAC brings the correct amino acid. That's the tRNA, the transfer RNA.
Transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit.
Okay, so that was actually a little bit further than I wanted to go. That started the translation. Really what I wanted to stop with today is just transcription. Transcription is just taking the original DNA strand, splitting it, and then creating an mRNA, a message from that split. All right? So listen, when we come back on Wednesday, we will review we'll review mRNA again in the message, and then we'll get more into translation, which is actually taking the message and translating it into the order of amino acids. Okay, so we're going to stop there, you guys. I'll stop recording.